fluid around the tumor environment, also called ascites. And there are a lot of papers that have been presented on that from a while ago. So once you go through this first step and you identify your biomarker candidates by looking at the changes in some biomarkers or proteins between these samples, then you can go into the verification stage where now you increase the number of samples and decrease the number of analytes that you're able to simultaneously detect. And you can also move this from the discovery sample matrices to blood in a larger population of people. And so blood's a little bit easier to get than tissue, or proximal, a lot easier to get than tissue and proximal fluids. And then you can begin to see if these changes that are being observed are significant and relevant, and if you're able to detect them with high precision and reproducibility. And then finally, what we're all sort of striving for here is application of these methods to clinical validation, which means, can a doctor look at the results and may make a diagnosis or use it in a diagnostic way for treatment? And this is an area that LCMS has not really flourished in yet. There are a few methods that are used in the clinic, but we're not here on a very large scale yet. And there are a lot of factors that affect that. And so some of this is what I'll be going over today. So how does discovery-driven proteomics different from hypothesis-driven proteomics? Well, here, hypothesis-driven is you have an idea based on some information of how much is changing in a particular target. And so you follow that target through different samples, different environments, and you see how it's changing, the extent and the reproducibility, the biological variation of that. And then ideally, when you start with your biomarker candidates, you want to make sure, is it found in blood? Is it higher in cancer? It's easier to detect a biomarker increase than it is to detect a decrease in samples. And so this is why we always usually try to look for something that is increasing in our samples, and then determine if these biomarkers are worth evaluating. So, whoops, moving on. How do we do, well, what's the difference actually between quantitative mass spectrometry and quantitative proteomics? So quantitative proteomics is when you're detecting the changes in as many peptides in a sample as possible, and then rolling them up and applying them to changes in the proteins that they come from. And so you're not really narrowing down your list, you're trying to look at everything. So you can imagine just if you're out in a field of flowers and you're looking at all the flowers and seeing which ones are taller and which ones are smaller. How does that compare to quantitative MS or targeted? In this case, you'd be looking at those field of flowers and you're like, just go look at the yellow ones. I'm going to ignore everything else. They're still there, but your focus is on a smaller subset of the peptide population. So the idea here is you're asking the question, how much of peptide X is in my sample? And then you can quantify it relative to a standard. We often use isotopically labeled peptide standards, which can be synthesized and purified when you know the amount you're spiking into the sample, and you look at the peak area difference or the difference in the signal between that internal standard and the analyte. The beauty here is the C13 and 15 labeled peptide elutes in the same place and behaves very similarly to the analyte, but it just has a different peak area based on what you're spiking in. And another option is, so that would be considered like a labeled analysis. If you're doing label free, you might have two different disease states or a control and a disease state. And now you're looking at that same peptide signal in two different samples that have been acquired differently in time. And you try to analyze those and see what the change in the signal would be. So this is relative quantitation or label free. So how do we do quantitative proteomics? There are a lot of ways we can do this. The labeling strategies are a little more expensive and intensive and could be limiting. So we've got metabolic labeling like SILAC, chemical labeling where we go in and chemically derivatize some kind of entity in the peptide or protein sequence. Label free is when we do not use labels and we have to analyze our samples one at a time, which is the expense here is often in the amount of time it takes to generate the data due to the need for additional replicates, or targeted proteomics. And so designing methods specifically to look at just a few smaller number of things than many, many thousands. So going into each of these individually to give you a background on what they entail. So stable isotope labeling with amino acids in cell culture, it has the acronym SILAC. This was originally presented on many years ago, and it's meant for use with growing cells in culture. So if you have a cell line and you can treat it with a drug, or you've got something that's resistant to a treatment, you can look at how the protein expressions are changing between these two cell states, and you feed them heavy media. So you give them heavy or labeled amino acids in cell culture, and they begin to incorporate those amino acids in their protein sequence. And so usually it is lysine or arginine that are incorporated in the media, and then those heavy amino acids are now included in the protein sequence. And when they're digested, they are also in the peptide sequence. And so here we've got the normal amino acids on the left, which would be light, and then media labeled with heavy amino acids, which would have the asterisk. And over time, so once these cells are labeled and you go to harvest the cells, you can combine them immediately. And so any sample prep steps that these cells go through, they are seen equally and will have the same level of variability. And this is one of the most ideal situations because you can combine your samples very early on in the process. And what you see from a mass spectral standpoint, or from the mass data, is you'll see doublets. So there will be the light peptide and the heavy peptide that's a certain mass to charge away, and you can quantify relative changes in these peak areas based on experimental parameters. So in order to do this, you need metabolic labeling reagents like mammalian cells. This is not like classical pulse chase experiments with radioactive isotopes, so perhaps a bit safer. And then the idea is you add these things into your media and you grow the cells. Then it can be done in a two state or a 3-state, depending on how many heavy amino acids you have access to. And this can all be controlled by budget as well as samples. But the idea is once you've grown all of your samples, you combine in that very first step, and then you do the reduction, alkylation, digestion, and then quantify by LCMS. And this is the kind of information you get where you'll see a light and heavy doublet in the 2 samples. So this represents the same exact peptide in pretty much the same exact abundance. And then for a 3-sample state, you've got the light and sort of the medium and the heavy in the mass spectrum. And you can pull this information out using software like Skyline to quantify the peak area underneath the chromatographic peaks. And so this quickly goes over the advantages and disadvantages of SILAC, where we have advantages, or the label is incorporated very far upstream and the variability is accounted for through this. The samples are combined early on, and the quantitation of most peptides is possible as long as you see the presence of the light and the heavy, or 2 out of the 3 if you're doing a 3-way. Unfortunately, this only works the way it's been designed with cell culture, and the media may be expensive, and only 2 or 3 samples can be compared at a time. And it is so technically this is incorrect. Human samples could be possible if you could feed people isotopically labeled food or water. And there was a study done some time ago with mice where they were fed heavy chow. But the process for this is a bit arduous, and the chow is expensive, and mice are not clean eaters. So they'll go in and the food will go around. So it's probably not something that people would want to do routinely. So another approach is using chemical derivatization, and this started many years ago with isotope-coded affinity tags, where you've got a thiol reactive group which reacts with your protein or peptides, usually the cysteine residue, and this can be modified to react with a number of different peptide reactive groups or protein reactive groups, like the N-terminus or primary amines. And then there's a linker region that you can incorporate heavy isotope like carbon-13 or nitrogen-15, or in some cases deuterium. This was very early on. And then the last portion is an affinity tag. So you can preferentially enrich anything that has been modified with this thiol reactive group. So the ICAT was used for targeting cysteine-containing peptides in digests, which not all proteins have. And the proteins that do have them usually don't have a ton. So you're getting some kind of subset of your entire proteome, and you're able to quantify changes on that level based on heavy versus light linker regions. So this has evolved over the years into iTrack and TMT, which is an isobaric labeling strategy. So on the top here, you would see a doublet, much like Skyline in the MS1 signal. You would see light and heavy. Down here at the bottom, iTrack and TMT differ because that once a peptide is labeled from across your samples, it'll have the same intact mass. And so the fragmentation is what allows quantification in the MS2 space. And how this is done is all of the versions of this light and heavy and peptide that's been labeled by these tags, if it has the same sequence, but a variety of different tags, the entire mass of that tag is the same. So these peptides from a bunch of different samples will all coalute with different mass to charge values. With the same mass to charge value at the same retention time. And then when the mass spec isolates it and fragments it, this reporter ion region happens very low in the mass to charge space, and it'll allow you to quantify changes. Each sample, each peptide from a different sample will have a different mass fragment here. And this mass has been extended now to like 130. It's much, much higher. So you can multiplex in a bunch of different ways. But this is how this looks, where we've got the isobaric chemical labeling. In this case, we have 4 samples, and they are digested, labeled, put over they're digested, labeled, put over a strong cation exchange to remove the reagents, C18 separation. And here you see the fragment ions in this region. And then from the bottom, you can see the MS2 getting you the peptide ID. And then you're able to sort of look at these peak areas and quantify the amount of peptide that's in the sample from each of these starting sample points. Using iTrack or TMT, again, as I mentioned, the precursor ion for the MS1 scan is the same for the peptides coming from all these different samples that have been labeled differently. And when the fragmentation occurs, this is just a blow up of the previous screen. You are able to identify the sequence of the peptide. So hopefully you could go in and then backtrack it to a protein of interest. And then this very small region, in this case, it's iTrack, is looking at the intensity of that fragment ion coming from each tag. So this would, the 114 is sample 1, 115 is sample 2, and 3 and 4. And you can see in this case, we would say, oh, the 116 or sample 3 has the most of this peptide in it compared to the other 3 samples. And then you're able to pull out the information from the peak areas for relative quantitation. Over the years, the number of samples that can be analyzed at the same time increases by modifying the tag itself, the reagent, and incorporating different isotopically labeled atoms to make the mass change different in this reporter ion region. So here, this is iTrack. We're looking at mass to charge of 113 up to 121. So this is an 8-plex that allows you to analyze 8 samples simultaneously in a single LCMS run. I think now we're up to like 20 or 21, and this requires high MS resolution because some of the mass differences in the tags are actually 0.1 mass to charge units. So you need to make sure these peaks are super narrow, highly resolved from one another in MS space, which usually requires high resolving power and an Orbitrap mass analyzer. So this is an example.
Couple questions in the chat. First question, Silac is not feasible with complex tissue lysates, let's say mice tissue lysate. Why can't you use Silac for mice tissue lysate?
Okay, so Silac requires the building and transcription of proteins while it's in the media. And it's usually, once you have tissue, it's usually a piece of tissue. And the biological activities going on there are going to be a lot different than if that tissue is like still attached to the living entity. So incorporation of those heavy amino acids into the protein sequence is not possible in a living being unless you're feeding them. So once tissue is extracted, whatever biological processes are going on would be difficult to relate to how it would actually be occurring in the living intact sample.
That's like kind of a follow-up. Isn't there a Skyline mouse available?
There may be these days. Actually, I haven't gone shopping. I don't tend to do the global proteomics in a routine basis. But yeah, I think anything could be available these days if you need it to be.
Yeah. And I know you had mentioned in the lecture, it's not Skylac because it's not cell culture, but Skylam in mammalian, like mice. You can buy heavy chow, and that effectively will label your mice, but the heavy chow is like super expensive. So if you ever want to like ask some of the gray hairs at ASMS, they will tell you, regale you with stories of picking heavy chow out of the mouse like litter and poop to recycle it because it's so expensive. And then we have a question on TMT. Is the advantage to TMT saving on runtime? Is that why you combine all the samples to save on runtime? Are there other advantages as well?
Yeah, so one of the major ones is exactly that. When you look at a TMT experiment versus label-free experiment, a TMT 10-plex allows you to look at 9 samples plus a control simultaneously in one run. You would probably want to analyze that in triplicate. Versus if you were to do those same 9 samples plus control in label-free, you would need to do each one of those runs in triplicate in order to get the technical variability of those measurements. And so there is like a balance. The TMT reagents are expensive if you're doing like a 1 versus 1 experiment, because you can achieve those by doing label-free by just the cost of the LCMS analysis. Whereas the reagents are expensive, or could be in the thousands of dollars. So if you buy a kit and you're analyzing 10 samples, now, yes, you are saving on that runtime by combining all your samples together. So there is a portion of like experimental design consideration based on your budget and then your LCMS availability. And when I was running a core facility, I would go through these calculations with people being like, oh, you only have 2 samples. It's easier to do label-free. And you'll get similar information, at least answering the questions that you want. Whereas if someone had 8 to 10 samples, then it would be cost effective to buy the kit and then just run the whole set in triplicate. But yes, that is a very good point. Thank you. Okay. So as I mentioned, when you get to higher plex levels, meaning more labeling reagents, now you can see the differences that you see in this labeling profile or the barcode are so close that if you don't have accurate, high enough mass resolving power, they'll come out as a single peak. So this is why using instruments that have a certain MS resolution is important to take into consideration. So time of flights and Orbitraps are the 2 most high resolving mass analyzers. But unlike Orbitraps, time of flights usually have an upper limit and you can't really adjust what the resolution is like you can on the Orbitrap. And so often TOFs are limited to 10 plexes or lower. Okay, and then for the instruments that can go to higher resolving power, you can see now, I think up here, this was showing 4 pairs of reporter ions that are close in M over Z, but now Thermo has developed these TMT kits where it's a lot more. And so you're able to, again, multiplex more samples all at the same time. And the importance of this is being able to measure your samples enough times to determine their biological variability and the technical variability in the measurements. And this allows you to tease apart what's being changed due to biology versus what's being changed due to just measurement precision. So for academic labs that don't have a lot of money and are looking for any ways to do some kind of quantitation, there are other less commonly used. So using heavy water is one where you can digest your sample in heavy water and this O18 atom is incorporated into the peptide during the trypsin digestion. Funnily enough, funnily, I don't know if that's a word, sorry. You can actually add O18 water to your sample after it's been digested and you add trypsin again, and it goes in and it'll still do that replacement shown in this chemical reaction here. So you can generate an O18 labeled standard and like have it. You can take also your same sample that was digested in trypsin, redigested in trypsin with O18 in one sample and regular water in the other, and you should be able to differentiate them based on this heavy oxygen that's incorporated into every peptide. So not so great because the mass to charge difference is only, or the mass difference is only 2. So usually it'll be a mass to charge difference of 1 if it's doubly charged. Another option is dimethylation. Again, not superly common, but if you're desperate, these are some things that can be used, and it's done post-digestion, reacts with the N-terminus of the peptides, and it'll impart a light and a heavy version. These are deuterated, which as long as you're not going above a certain number of deuterium, like 4 to 6, they'll still have the same chromatographic properties as the non-deuterated reagent. So this just goes over some advantages and disadvantages of the chemical labeling, where advantages are we can quantify in the same LCMS run when you're doing chemical labeling because their mass to charge is different, so you can combine them before separating by LCMS. This can be done on human samples like plasma, tissue, animal tissue, and so on. Usually there are kits available depending on the goals of your experiment. There are also these quote unquote, cost-effective alternatives that can be more laborious just for that. And multiplexing is available. So disadvantages are that the label is usually incorporated post-digest. So there are a lot of variability imparted in the steps leading up to that. They're usually not combined until far into the workflow. The kits can be expensive if you get the good isotopically labeled ones. Labeling from anything may not be 100%, so that's something to keep into mind. Data analysis software is necessary, and peptide IDs are not guaranteed. So often you can get quantification of peptides from iTrack and so on, but you might not know what their precise peptide sequence is, and therefore how it dials back up to the protein level. Okay, so the last
more. Oh, yeah. Sorry. Couple questions in the chat regarding labeling. So a question about iTrack. What is the point of making isobars? Why do isobars elute at the same retention time?
So the purpose there is, I don't know, here, this is an example of the MS scan for the precursor ion. This is not just one peptide. This is 1, 2, 3 peptide, the same peptide sequence from 4 different samples that were combined with that chemical labeling reagent. And in order for us to figure out what this peptide is, it needs to be isolated and fragmented. So in this case, you want all of the different samples with that same peptide to be isolated simultaneously. So they have these tags that have, the total same mass value. They've got the reporter ions, which differentiate by mass to charge. And then you have the balance, which helps you to total up for each reactive reagent the same total, so that they will elute in the same place and have the same mass to charge. Then when you isolate them and fragment them, the only things you're going to see are what's coming out of the peptide sequence. So you can ID it, and then this reporter mass region. And so this is where we've isolated this 791, whatever peptide this is, with the 4 different labeling reagents on it. And then we can see all of the Y product ions and some of the B, so we can identify it in the MS2. But this reporter region in chart C that's blown up, this is how we quantify from each of those 4 samples how much of that peptide is present. And so we can do it on an 8-plex. Presumably one of these channels might be a control, so you could normalize all of these signals to a control channel. And so that hopefully it's the idea is these intensities give you a quantitative measurement of these peptides in the complex samples. Does that answer it? Is that clear?
Got a follow-up question on TMT. Is fractionation always necessary for TMT combined samples?
So fractionation, which I haven't gotten into yet, that is something that allows you to take your one sample and turn it into a lot of other samples. And so some people will fractionate to get, it's so what the idea is, what it does is you take your sample, let's just say for argument's sake, you split it into 8 fractions. And by doing this, you're separating your peptides on some orthogonal means. So you're not necessarily using acidic reverse phase, which is what we use for LCMS. You might be using basic reverse phase, so high pH, which separates the peptides in different orders. Or you might be using strong cation exchange, which separates the peptides based on their cationic properties, or or I think those are the most commonly used ones, but those are the phases that people will use. And then they'll collect different fractions along the chromatographic timeline. And then each of those fractions will be then analyzed by C18 acidic C18 reverse phase LCMS. The idea there is you're taking a portion of your sample, your whole sample, and separating it from the rest of your sample, and then analyzing it individually over a reverse phase gradient. And the idea here is you are able to detect less abundant species. And so you're improving your dynamic range of peptides that can be detected from your sample. The problem is reproducibility of that fractionation and quantifying some of the signal if it's been split into a couple different fractions. And so it depends on your experimental goals. If you don't fractionate and you detect all the peptides and proteins that you may have a hypothesis for, awesome, then you don't have to fractionate. Some people will fractionate into 8 or 12 or 96 and then maybe back off the fractionation level once they figure out what they need to see in their sample or make biological relevance of it.
So many questions coming in now. Okay. Maybe this one's a quick question. For disulfide or modified cysteine residues, can you still label those?
No. Usually the interaction of not disulfide. So if a cysteine is cross-linked with another cysteine, you usually can reduce those and making free thiols, which then should be reactive with other things. But if cysteine already has been modified or like methylated, so I'm not an expert in like the full nuanced chemistry. If that reaction is reversible and you can reduce it, then yes, it should be fine. But if it's not reversible, then obviously that site is now blocked and you can't label it.
We still have a couple more questions. I'm going to do just one for now, and then we'll take the rest later and let you keep going. Okay. Okay. But maybe relevant question. Do these labeled quant use a top N DDA?
So they can, yeah, I mean, they can, and there's a lot of flexibility to the experimental design. When looking at an iTrack or TMT labeled sample, you can define the top N, meaning it's going to do MS1, look at all the peaks that are observed, and then pick the top N number, whether it's 10 or 20 or whatever, and isolate those to do MS2 on them. And that will give you the peptide sequence plus the quantitative aspect. What top N you pick depends on your chromatogram, your peak width, and being able to sample as many of the chromatographic peaks as you can without oversampling, meaning measuring the same one over and over again, or undersampling and skipping ones that are maybe too not intense enough. Does that answer that, hopefully?
I think that answers questions. We've got a couple more questions, but I'll hold them so that we can keep going. And I see some proteomicists, mass spectrometrists in the chat that are jumping in to answer some questions. That's fantastic. Please keep doing that. Drop some references, drop your experiences. We're just two people with our own backgrounds, but we know that there's a lot of you out there that are also doing mass spec all the time. Definitely keep talking amongst yourselves, but we're going to continue with the lecture portion.
Thank you. Okay, so the advantages, I think we're on to label-free. So label-free proteomics is often a great place to start because it allows you to see gross changes, large changes between samples And if those are the changes you need to see and make sense from a biological standpoint, that should be good enough. But often you're limited because you might want to be looking at hundreds of samples, and that would take a long time from the label-free strategy. So here the idea is often called differential proteomics. No labeling is used. Samples are run at different times. So while you may be able to batch process them, like in a 96-well plate where all of the digestion happens practically simultaneously, you're analyzing them by LC-MS at different times, one run after the other. The abundance of the peptides you observe are derived from usually the MS1 signal, because you are not frequently and repeatedly fragmenting the same peptides over and over, unless you're doing data independent acquisition, which Lindsay is going to cover in the second half of today. Ideally, necessarily, multiple replicates should be run of each sample in random order so that you are 100% sure or very high percentage sure that the changes that you see are related to biology and not a poor injection or bad electrospray. So you want to make sure the technical variability is assessed by doing replicates. And then you want a quantitation is assessed from a number of varieties of options. Spectral counting is one, spectral intensity, extracting the MS1 or the MS-MS signal if you're doing DIA. And that basically is how that works. So an example here from a label-free experiment, we ran the same normal sample in red 4 times and the same tumor sample in blue 4 times. And because of the nature of LC-MS, particularly using nanoflow, often you get retention time shifts. So that doesn't show the alignment, but often the data can be aligned by looking at retention time and mass to charge features so that they can overlap. Once they're aligned, then you can begin to pull out individual peptide data from the MS spectra. So here's an example doing quantitation based on the precursor where we've got the 4 blue replicates, the 4 red replicates. MS scans were taken at the little diamonds and MS events were taken with these big triangles. And we're able to get a ratio based on the peak area of the average of the blue, the average of the red. And then from all of these, you might only get good MS2 spectra for 1 or 2 of these individual runs, but if it aligns with the mass to charge and the retention time and the charge, then you may be able to quantify and just assume it's the same peptide. It's not always the case, but this software gave information to allow some of these to be made, some of these assumptions to be made. So which are the best options? Some people have already started asking these questions, like what are the considerations for designing targeted experiments? Can you quantify by MS? What is the best mass analyzer, if you even have a choice? And then how do you evaluate your data? So the goals of moving from whole global quantitative proteomics to targeted proteomics is you need to understand what answers you need to get for the questions that you have. Are you simply looking to detect something and see if it's present or not detected? Or are you trying to quantify discrete changes in samples, in which case your number of figures of merit, like accuracy and precision and linearity and things, you need the information to calculate these. So this is just a laundry list of considerations in developing a targeted assay. And I'm not going to go through it in total detail because I'm sure people will have just better questions that these we'll get to eventually. But the idea here is, why are you targeting a sample? Do you have empirical evidence based on Western blots, based on ELISAs, based on mass spectrometry? Or is it like a hypothesis like, oh, this would make biological sense? Any of those are valid. And it's always good to try to go in with the hypothesis if possible. I mean, it's interesting to think about what changes cancer may cause, but you can kind of dial into the types of proteins and interactions that are going on with cancer. You may not get it right by guessing 100% of the time, but you talk to a good biologist or cancer researcher, and they probably already have their list of favorite proteins. You also want to think about what amount of the protein might be present in the sample. So we know currently we can measure about 100 or so, maybe less attomoles of a peptide in about a microgram of sample when doing nanoflow on conventional LC-MS. That's without enrichment, that's without fractionating. So how much of your starting material do you need to get about 100 attomoles of protein? There are a lot of assumptions of the targeted protein. Sorry, there are a lot of assumptions here, and we're glossing over things like sample loss and digestion efficiency, but this is a ballpark starting point. If a protein that you're looking at requires 10 milligrams of protein, then that's going to be really hard to do without significant sample prep and enrichment. How many targets do you want to look at per sample? And then how many transitions per peptide, because this might control what type of instrument you use, a high resolution mass spectrometer or a triple quad. And then cost. Do you have money to spend on labeling reagents or internal standards, or for lengthy LC-MS runs, because those often come at a cost too. And then how much time do you have to spend looking at sample acquisition and data analysis time? And then how unique is the peptide or protein? That's something you can figure out ahead of time by doing literature search and blast searches. And then what are the potential for false positives and interferences? So this is something I just tell everybody when I do these kind of things. Never underestimate the literature. You can sit at your computer and figure out a whole bunch of things about what you might be looking at before you actually look at it. And this really gets down to, sometimes things haven't been published. For example, quantifying a certain protein in a certain disease state, not just because they haven't been thought of or tested, but because there were no positive results or it was just impossible. And I've encountered this a few times in my scientific career. And so that's just something to consider. If you're going to do a literature search, you're like, oh my gosh, there's no information, I'll be the first to publish. Maybe you won't, because maybe it's impossible to get the data you need to make a significant impact. Also, you can look in the literature to see right now, like I had mentioned earlier, we know about our detection limit for peptides in a digested sample by LC-MS, give or take. And these are all things that are usually plasma proteins. If you're looking at plasma, high abundance, perhaps not that interesting. And in order for you to get down to lower concentration peptides and proteins, you have to do something to the sample. And so this is a really cool figure, I think, that gets into the different levels of things you can do to your sample and the impact it has. So when we do LC-MS of peptides for proteomics experiments, we obviously start with a protein sample, which could be cell lysate, tissue, plasma, any other kind of biological fluid. We usually start with 10 to 20 microliters of plasma, or a little over 1 million cells per sample. This can be dialed down, obviously, now that we're getting to more sensitive instruments, but this is just in general a good amount to have when you first start. You can do protein level enrichment by amino affinity. You can do an anti-protein antibody to enrich just your protein. You can pull out particular aspects of the proteins in your sample. You could do cellular fractionation, you could do size exclusion, you could do ion exchange, and separate your total number of proteins into bins, and then fractionate them analyze them one by one to help get to lower amounts of protein per sample. Another fractionation step is at the peptide level. So after you digest, now you can pull out your phosphorylated peptides through IMAC. You can do amino affinity by anti-peptide antibodies if you have that capability, or you can do fractionation, like I mentioned, cation exchange or basic reverse phase. Now, this step here, this nano LC, had been the major workaround most researchers use to get to that level of sensitivity. You can load less sample and detect more if you're running at a lower flow rate into the LC-MS. And for the quantitation portion, often triple quads are used, but also using a targeted list on a high resolution accurate mass instrument will work too. And this allows you to see the whole complement of product ions rather than just picking out certain transitions. This sample enrichment process, I'm just walking through some numbers with you, starting with plasma, just so you get an idea of what they do. So plasma is about 50 to 70 milligrams per milliliter of protein content. So if we want to analyze only a microgram, it winds up translating to 15 nanoliters of plasma. Obviously, we can't work with such a small volume, so we start with more and then dilute before the analysis. But if we were to do a protein level enrichment, so for plasma, let's take away the top 14 most abundant proteins to look at the more interesting ones. If we bring that sample after depletion back to that same starting material volume, it's about 220 nanoliters of neat plasma equivalent per injection. If we do the orthogonal peptide level chromatography, now it looks like the equivalent of what we would be loading if we hadn't done the enrichment is between 500 and 1500 nanoliters. So you see, even though we can't load that much on a nano column, the equivalent we're able to see gets us down to proteins that are so low abundant, we normally wouldn't detect them without this level of enrichment. And then antibody enrichment, like anti-peptide antibodies, allow us to just pull out those peptides and ones super similar to them, which is like a tiny little subset of the whole sample. And this can translate to starting with a volume of a milliliter per plasma, and then taking all of that and injecting onto the column. So this is the effect that the different levels of fractionation have, and kind of understanding where you need to go will help drive that. Also, the more separation processes you put your sample through, usually the better signal to noise you have. And this is due to a relationship of, with every mode or stage of analysis or stage of separation, like 2 is LCMS, GCMS, so you're just separating and then analyzing, or MSMS, fragmenting and looking at those data. Unfortunately, there's signal loss associated with increased stages of analysis, but the noise usually goes down quicker. So these are just examples of how you can hyphenate different modes of separation, all amenable to one another, to get to improved signal to noise for your detection, which is ultimately based on this selectivity idea. So just for those that are like, I hate nanoflow, but the reason that we go to it is looking at this sensitivity improvement on the y-axis. So this is looking at flowing at a higher flow, starting with a 2.1 millimeter scale. So you can load a ton of protein onto that. But if you take and dial the flow rate back and adjust the column inner diameter from 2.1 to 1 to 300 microns to 150 to 75, what you see is this gain in sensitivity, this gain in signal to noise. And this allows you to have better quality data and confidence in what you're doing for identifying those proteins and quantifying low abundance ones. So if anyone just ever is like, why don't we do high flow? High flow is great, and you can bang your samples through in a short period of time, but often you're giving up the sensitivity in certain analytes that you wouldn't detect otherwise. So this is just a little history of how people have been quantifying by using MS as a mass analyzer and detector. And this is nothing recent. It started a long time ago. And you can see over the decades, these are the 2002 was SCILAC, 1999 was the ICAT, iTrack came about from there. But inherently, I would say, as being on Team Mass Spec, mass spectrometry is not inherently quantitative. There are a lot of things that affect it, and there are some unknowns in our calculations for being able to quantify things. So if we compare this to Beer's law, which is for spectroscopy, we get an absorbance from a signal, and we have a known extinction coefficient we can usually calculate based on the properties of the molecule we're looking at, and a concentration which can be measured based on that signal and predetermined extinction coefficient, and then a known path length. So all of these things at any given point can be set up so that there's only one variable, and we can measure the rest, or the rest are known, and calculate that one variable. Here on the MS side, we've got analogous, some signal, but the concentration is affected and has this ionization coefficient or transmission coefficient or something that affects not just maybe the concentration of the analyte, but the concentration of the sample that it's in. How many other ions are being produced at the same time, and what is their effect? And this K value is largely unknown unless you go through and try to empirically determine it. So it's hard for us to do something like change the concentration because we don't know if this ionization coefficient is changing at the same time or not. And making assumptions often makes you just have wrongly interpreted data. And so this is where using the labels helps us to sort of overcome this ionization issue, particularly if we use labels that are for the same peptide, but label them a little bit differently. So this is one example where we use stable isotope dilution, and we have these amino acids in the peptides we're looking at. Trypsin clips at lysine and arginine on the C terminus. And so the majority of the peptides in our samples digested with trypsin have lysine or arginine on the C terminus. We can take and label the carbons and the nitrogens in these amino acids and synthesize peptides for the peptides we want to target, and then spike them in known quantities. And so this kind of came together in the mid 2000s and made it really cool because it just worked. Then when we take the peptides and we fragment them, we tend to fragment across the amide bond next to the carbonyl and this nitrogen, and we generate B ions where the charge is retained on the N terminus, and Y ions where the charges are retained on the C terminus. And these numbers allow us to assign fragments of these peptides starting from the N terminus or the C terminus and add up the amino acids to look at the whole mass and see a loss of an amino acid, and then you know what amino acid was lost based on its mass. So this allows us to go through the sequencing of each peptide. And this is just an example of a light and a heavy peptide where the phenylalanine, in this case, this is super old. I was in grad school last decade. Anyway, this phenylalanine was the heavy labeled version. And so the fragments here shown in red, the light is 1031 and here 1037. We had 6 carbon 13s on that phenylalanine, and that's incorporated into the B ions. The Y ions here don't have a change in mass because this would be just the proline and lysine, which are not isotopically labeled. Now we usually label these C terminus, so all of the Y ions that we see have that signature mass shift, and we can quantify it on all of the Y ions. Okay. This is just kind of reviewing before looking at stable isotope dilution. You spike in your standard in a known amount and then quantify the peak area relative to the light peptide, which is coming from your sample. And this is how we often use Skyline to do this. And the idea here also is quantitatively, you should see a linear response for changes. So we look at often the ratio of the MS response of the analyte divided by the internal standard, and the ratio of the amount of these that if you spiked in a fixed amount of internal standard and a varying amount of light, then you should also see when we do the light versus the internal standard, this slope with a regression line of y equals mx plus b or y equals x, because usually in this case, the slope should be ideally 1 and the b, the background signal, should ideally be 0, but that's often not the case. So we like to generate these lines and look at the slope and the y-intercept to see if they have any significant impact. Here, one question people may have is like, oh, I don't want to analyze, I don't want to generate peptides with like a individual internal standards for all of my peptides because it's expensive. And so if we take a step back and look at the impact of what having an internal standard versus label free, and then an intermediate, which is called a labeled reference peptide option. So this was a response curve that was generated all like individual concentration points, obviously, and these are plotted as measured concentration in femtomole per microliter of that peptide to theoretical concentration, what we think we actually spiked in. And here on the left-hand side, we had an internal standard. So the measured concentration is that analyte divided by the internal standard. multiplied by the amount of heavy that was spiked in, and this gives us the femtomole per microliter concentration. Then when it's plotted, we look at the R squared and the slope, and that's all fine. But the coefficient of variation, which means the variability of each of the measurements taken, is about 1.2%. So we're able to get very reproducible measurements when we normalize a peptide by its internal standard, same sequence, just internal standard. If instead we take these same data that were generated here for the light curve, but instead of dividing by the isotopically labeled peptide that we're looking at, we use a different labeled reference peptide that appears somewhere else in the chromatogram. So it's a different sequence, a different retention time, different ionization potentials, different fragmentation. But we can use the same one peptide internal labeled reference for all of our peptides. What we see here is when we plot that slope, our coefficient of variation on these data points is now 5% instead of 1.2. And so we also see there's variability down at the low end and at the high end that this labeled reference peptide can't account for because it's occurring somewhere else in the analysis. And then last, label free. So let's just say we're targeting a bunch of peptides and we don't have an internal standard. We're not doing any kind of normalization. This is the same exact data that was in A and B. So we're looking at the same peak areas of the light peptide, but now they're not normalized. And now we see for these replicates, there are data points that are dropping down, and perhaps we don't know why. Perhaps there was like one replicate was just not so good. And because we can't normalize and take that variability away, now the coefficient of variation is up to 13% or 0.13. And so understanding that this is all the same light data, the impact that having an internal standard has is accounting for that variability and improving the accuracy. Okay, so this goes through some questions of like, what do you want to target? Do you want to go through and have empirical LCMS data first? Do you start with pure protein standards? Do you synthesize peptides? Are you getting your protein targets from the literature? And then how is that literature data produced? Do you have access to it? Can you look at it and see how this will influence your experimental design? So it's always good if there is data to consult it, particularly if you can't generate your own right away. And peptides, they fragment similarly, even on the different mass analyzers. So, and this is just based on if you're using the same type of fragmentation mechanism, meaning collision induced or collisionally activated dissociation, you should see a lot of the same fragments, perhaps not at the same ratios, but you can be pretty confident that they will be generated similarly. So now, if you have empirical data, what do? We search the data against proteomic libraries. You can identify peptides with certain confidence. You can generate a list of these peptides, like the ones you're most likely to detect, and then target them. Whoops. There are some resources you can use to sort of seek out this information. There's a lot of publicly available databases. Panorama is a great one for looking at targeted workflows. Passport was generated a while ago, and this looks at the most intensely observed peptides after a tryptic digest of a particular protein. So this is based on empirical information. Point being is you would want to target peptides on the left-hand side of this plot rather than on the right-hand side of this plot, particularly if you are not seeking out post-translational modifications or certain site-specific modifications. And then there are peptide and protein targets from public resources like the CPTAC assay portal. And this goes down into the peptide sequence from a particular protein, limit of detection and quantitation, precision and repeatability that have been achieved empirically. If you have no idea where to start, you can do in silico digestion and you can generate the data and you can generate in silico versions of the targets that you would be looking at. So you start with the general rules, and these are some settings in Skyline that you can set up for doing digestion and looking at numbers of miscleavages, what the optimal peptide length is, make sure it's not close to the end terminus of the protein because you might have exoproteases that are going in and chomping up your protein. Generally, we like to exclude labile amino acids like methionine because it's prone to benchtop oxidation, which means now your peptide is split into the non-oxidized methionine and the oxidized methionine, which is different by mass to charge and often retention time. So point being to beware of post-translational modifications. You can usually look up and see if your protein may have glycosylation or phosphorylation or something based on a consensus sequence or something in there where it's been determined to exist. So here's where we can go into some examples of how to use Skyline, data dependent, running a database search and identifying peptides and then generating that spectral library in Skyline. So this is kind of what we did with the plasma data set that you should be able to download. From here, we can select a certain number of targets based on what we want to track in a number of samples and go through and sort of refine this process. So for people using triple quads or SRM, we often want to take the total number of fragment ions and narrow it down to like 3 or 5 most intense that may not be interfered with. And this requires a little method development, but once those are set, they're pretty good. For PRM or parallel reaction monitoring, you can keep all of those and you can just worry about how the data looks and then not include some of those peptides at the end, but they will always be collected by the mass spectrometer. And ultimately, by going through this process, you have a refined transition list for method optimization. So to guide you through some of the, I guess, hiccups or things where you're like, oh, this is my first time doing this, I don't know what to do. So this just walks through some examples in Skyline of data that we generated, and we're looking at the MSMS spectrum. So when I set up a list and I'm looking at like, what's the best peptide to pick from proteins, I will usually look at the +2 charge state and the +3 charge state of a particular peptide. So here on the left-hand side in what we call the tree view, we've got the protein accession number, the peptide sequence with it, which is EHVAV, ... And then the +2 charge state at 617 and the +3 at 412. And then we've got a bunch of product ions for each one. These were all automatically generated in Skyline by giving it a certain number of criteria, like give me 4 or 5 transitions, give me the 2 charge states. And so that's what we see. Then when the spectral libraries are brought in, we can observe them this way, visualize them plotted. You can have Skyline label all the peaks that are observed. So these were already identified by a spectral database search. And then that result from that spectral database search was brought into Skyline. And now we're able to see these peptides. So we see unreacted precursor in the 2+ charge. We see a whole bunch of product ions. It looks pretty good. But we also see information for the +3. You're like, oh, well, at a quick look, you're like, well, the +3 isn't generating so many product ions. So maybe I don't want to pick that. But if you look at the scale on the +2 and the +3, the +3 is 1 million and more signal, whereas we are at 1 million and less for the +2, even though it generates more product ions. And so this is really one of those things where I don't know what to do. Initially, I might keep both of these. And then when I wind up getting it into a actual targeted workflow, get rid of one of the charge states. But for right now, we often like to try to pick higher mass to charge fragment ions because you can see up here, there's not a lot of chemical noise that's coming through with these signals. Whereas if we had to pick the charge 3, we would be picking ions down here where there are a bunch of little gray unlabeled peaks that are going to be generated that have nothing to do with our peptide and may increase the noise of our sample signal, thus decreasing the signal to noise ratio. But again, I often keep a bunch of them in until my last phase where I might delete one of these charge states. So also some peptides are super beautiful. So when we go through, we're like, this is a lot of ions that are generated. This looks great. I'm pretty confident that this is the peptide. But some peptides don't look great. And so even though this was identified based on database search, and it got whatever score based on my cutoff criteria, it's not generating ions that I think are worth pursuing. If there were other peptides from this protein, I might just delete this one and not pursue it. But the point being is not all the data you get are perfect, and you might be left scratching your head with like, why was this identified? It's not, doesn't look that great. But for the best results, you want to explore the data to understand it. Use predictions, algorithms, literature as your guide, and then also check the data manually. You might see great, you know, this peptide might have come through as like, oh, this is a great hit, but you wouldn't know really what it looked like until you looked at the MS2 scans, which are not often presented in a proteomics experiment after you do the database search. And then once you do this, like every step, every experiment you do, you'll become experienced in generating these targeted methods and become more confident in your data. And just a quick little analogy, there's this, you know, little story about different people touching an elephant, touching a different part and thinking it's something different, like this when they're all blindfolded. So this one person's looking at the leg saying it's a tree, one's touching the trunk saying it's a snake, one's saying it's a spear. And it really just depends on your perspective and how much information you're able to take in. So if you're able to look at everything, you might be able to identify it as one kind of elephant or another kind of elephant. Mr. Snuffleup, I guess, has no ears, so that would be incorrect, even though you might have identified it as an elephant because it has a trunk. Or, you know, sometimes the data are clear enough where you're just not identifying something. So let's see, I'm going to pause there and see if we have questions before going into the next phase.
We do have a ton of questions. I guess, how are you feeling for time? I think we've got about 20 more minutes. Is that right? Let me check.
Yeah, so here, what I was thinking is we've got the hands-on, and we can do the hands-on, and we can go through kind of individually. Or if people are not wanting to do that, we can go through just additional data and I can walk you through how to do some things without doing it live. So it's more of a, what do people feel?
How are people feeling? Maybe a couple, how are people feeling? Drop some feelings in the chat. Continue lecture or go to hands-on? Do it live. Hands-on, hands-on, hands-on. Continue, okay. Live, hands-on, hands-on. Getting a lot of votes for hands-on.
Seems like we've got some hands-on. So has everybody opened up, downloaded their plasma proteomics thing from the websites that I posted? Tutorial.
We did have a couple people say that they were having problems downloading. I don't think it's on our end. I think it might be some antiviral. Maybe some people are running like an antivirus for their computers, and maybe that's blocking the download, because I think other people have had no problems.
Okay. Hold on, I've got to extract. So I hold on a second. Let me share just so y'all can go through this with me. So I just downloaded this from the website and I got a zip file, and I'm just going to go extract all. I'm going to pick something. I'm going to pick my documents folder and we'll just put it in there. Extract. Okay, so in my documents folder, I've got a plasma directory. So I haven't renamed or done anything. Hopefully y'all can get along this far. And then there's also this plasma tutorial, which is the PDF that walks you through everything. That's not it. Let me get rid of that. No one needs to see that. So I'm just opening up the plasma tutorial now. I am sharing, right? Can everybody see?
Yep, we can see the PDF.
Okay. So here, just to walk through some, well, let's go back. The idea here is we've got 6 proteins that we're going to be looking at, and all of the materials you need are in this plasma directory for this workshop. Lindsay, you made this tutorial up, right?
Correct.
Thank you. Everybody thank Lindsay. So if we go, yeah, I think we'll first, we assume we know nothing about these 6 proteins. And that'll be the first way we're going to start off, because I'm assuming nobody knows anything about these proteins. So you can build an assay without any prior knowledge, because you're going in and you're looking at information about the IDs, you've got your identifiers. So I think, if I'm right, we have, this is how you would get the FASTA files for your proteins and load the sequences into Skyline, because Skyline needs that information in order to generate peptides from the protein sequences, or you give it a list of already clipped peptides from the digestion process. All right, Sophia, you have raised your hand. If you want to come off mute and tell me what's going on, we could do that. Nope, the hand's down. Okay, maybe it was an accident. Everybody so far kind of going here. I would say, if we go to this plasma folder, we can, there's a plasma standards Skyline setup that should already, you have to extract this. Make sure you extract this before you just go into a zipped directory to open it up. But you should be able to go in and open the Skyline document by double clicking. All right, going to remind me later. Then we've got this Skyline file. Now this has been pre-populated for all of you with the protein accession number and the protein sort of code name, and then peptides that have been filtered based on digestion settings. So what I'm going to show you first is, if you hover over this, if you hover with your mouse, you click it and you hover. Nope, you click it. Oh yeah, see, so this window pops up that has all the information that Skyline has pulled from the FASTA file. So the FASTA file has the protein name, accession number, preferred name, the gene code, the species it came from, and then the long text description. And then it has the sequence of the protein. So one thing I want to point out first is this one has an N-terminal methionine, which is usually the signal sequence, and often proteins are processed after translation and have this methionine removed. If you're not sure of what state this protein is in, I would say don't target any peptide that includes that first amino acid in the N-terminus. And that's why when we go through the in silico settings of like peptide settings, so settings, peptide settings, digestion, you can include different proteases. You can include maxed miscleavages. I usually have 1 or 2. In the prediction tab, nope, I don't want to do that yet. In the filter, this is looking at the minimum and maximum peptide lengths, and then exclude the N-terminal amino acids. So here's where I would put in like at least a few amino acids to exclude at the N-terminus. Okay, everybody good so far? All right. And then for those of you that haven't seen Skyline previously, if you click on that protein name like I have, what that's showing me, excuse me, is it's highlighting all of the peptides from that ApoA1 that have been detected in the chromatogram. So if I left click and zoom in, I can get a bit of a better view of these peptides, and it's labeling them based on the first 3 amino acids or however many is needed to make it a unique peptide ID. And we can see these are all nicely separated. That looks great. If we click on the first peptide that has the DLA sequence, what's shown here is a couple of peaks in the chromatogram where something has been picked at 9.18 minutes, but there are some other peaks here. So something I like to do is like, let's zoom in, again, left click, or if you right click, you can do auto zoom X axis and make it best peak. And that'll zoom into the peak that Skyline thinks is your peptide. So what's nice about this is we see the retention time, we see the signals from all of the product ions. If we right click, we can put in, hold on, where's properties? Legends. We put the legend in, we can see the product ion signals that are being extracted from the data and their different colors. And then if we click on each of the individual product ions, it'll highlight them. And so normally if we were doing this live and we had the raw data and we had to import it, you would see how many points across the chromatographic peak are being counted. That's what this little red question mark is. Unfortunately, sometimes when you zip and transfer Skyline files with the raw files, that connection, depending on how you unzip it and load it up, can be lost. So this one isn't registering right now, but you could go through each of the peptides and see how they look. Also, these peak area indicators, these little green balls, they're showing you that there is signal for each of these product ions. And more than 50% of them have been detected. So that's a green signal for the precursor. Even though it's not actually measuring the precursor, all of these signals are kind of give us a confidence of what's being detected. And so that peptide too. If we get to a point where there's no peak area indicators, like let's go to ApoA2. There's no data here. That's because we're not actually collecting data in that one file. So now I'm going to move to the ApoA2 tab. So this is the ApoA2 data file. And now we're collecting peptides from ApoA2 in this run, but not ApoA1. Let's see. What else can we do? If we click on edit view, modifications, protein, we don't want that. So let's go through some of this other information right now. So also, those of you that are new to Skyline, I have these data should probably be pretty standard when everybody opens it up, set up. I have the chromatograms from my individual protein runs on the lower portion. On the upper portion, I have the retention time plot. So there are a lot of different retention time things you can look at. If you go to view retention times, I can look at the peptide comparison. And so this is adding a tab to that area, and it's looking at all the different retention times on the y-axis and all the different peptides on the x-axis. Then I can go through and sort. Right now it's sorted based on that peptide's location in the document on the left-hand side. So if I sort it based on retention time, now I see that my earliest eluting peptide is this first one, and I should be able to click on it. And it'll highlight it in the left-hand side. And it's this peptide SQAIA. It's in a different protein. We can kind of go figure out which protein it's in by clicking. There we go. It's in the ApoB. All right, and that's the nice peak area. Oh, it's also shown right here. Okay, we can also sort these based on, whoops, peak area. That's more random because now we're looking at the retention time based on peak area, and we would expect the peptides to just be all up and down based on their peak area. In the peak area plot, if we go to view, there are also a lot of different ways it can be plotted where for the replicate comparison, this is the view that we're observing right now. It's most ideally used when you are targeting the same peptide over a number of runs and you want to see how it changes over a number of runs. In this particular Skyline file, every run is for a different protein. So this view is not so useful for us in this setup. So if we look at peptide comparison, now we can look at all the peptides. Let's see. For the different runs, and how they compare based on peak area. So I don't know if we have to like zoom in here. Yeah, so if we zoom in, or we could put this on a log scale, you can see all of the log peak areas on the y-axis and see how their intensities change with each of the peptides. And so if you click on a peptide, it'll bring you to that peptide, and we just need to go to the proper protein to see the signal. There it is. All right. Does anyone have questions on things they want to get from this data set but haven't figured out yet, or similar things they want to get from their own data sets that we can use this for, and we can kind of get a couple specific questions hammered out?
Got lots of questions in the chat, but only one of them is maybe regarding this data set. There's a question asking, what exactly is a scheduling window and what do you use it for?
Oh, okay, that one I can show. So a scheduling window is shown here. And if you think about when you target a list of peptides and you have 5, it's pretty easy for a mass spectrometer to, one right after the other, target each of those 5 in like a cycle. And like 1, 2, 3, 4, 5, go back to the top, 1, 2, 3, 4, 5, and you'll get enough chromatographic points across your peak. Even if the peptide's not there, it's not spending a whole lot of time on each individual peptide. And so not a big deal when you have a low number of peptides to target. Once you have a high number of peptides to target, like 100 or 1,000, that means your instrument's going to spend so much time in a given cycle on peptides that are not actively eluting, and you might only get a couple of points across your peak, because let's just say, for argument's sake, it takes like 10 milliseconds per each of these transitions. The more you have, the more time it's going to spend in a given cycle. So what you do is you schedule your peptides. And I think I actually have, I think I have a plot that kind of gets to this here, because this is one of the things. So this is looking at, can everybody see? Oh, here we go. So why do you want to schedule? We can't always detect all of the peptides in a given cycle, so we need to split them up to when they're eluting, and spend more time on peptides when they are eluting. And we get the elution information from discovery and from setting up method development and optimization runs. So let's just say we've got this one peptide at 28.3 minutes, and we're only going to collect it as it elutes, and we're going to put a little window around it. Now, retention times shift, like what this is showing, as we do our runs, the peptide's moving earlier and earlier, and we start having it go outside the retention time window where we've programmed the MS to look for it. And so we want to set these windows as wide as we can without spending too much time waiting for a peak. And so these plots are what give us that idea of, if we put a 10-minute retention time window around each of the transitions, and we have 325 transitions, this is going to spend a whole lot of time generating data for transitions that are not actively eluting. And so we're kind of looking at the scheduled time here and the number of concurrent transitions. Each of these transitions, if I said it takes 10 milliseconds to acquire, then 10 milliseconds times 100 transitions is 1 second. So that means that this 2-minute retention time window, we're at about a 1 second cycle time to target 80 transitions here. And if we go longer, then that means we're going to get fewer data points across our chromatographic peak with that same dwell time. And so what we try to do is set these parameters up so we can see how many concurrent transitions there are, or precursors if you're doing PRM, and making sure that we're spending the right amount of time on these as they're eluting. So if we look at the Skyline file here, this is looking at concurrent transitions, and we could set a 2 or 4 minute, or even a 5-minute retention time window around these peptides for this run, and we would still be able to get enough points across our chromatographic peak. Okay. If I can go through the questions, how does utilizing multiplexing influence detection of low-level peptides? So if you use multiplexing and that peptide is not present in all of your samples that you've combined, or it's very low abundance in some of your samples, but high in perhaps the carrier channel, you will be able to still identify it. And then the quantification from the reporter ion will allow you to get a sense of where it stands relative to all of the other samples that you're multiplexing with. If you did label free, it may not even have been triggered for MSMS, so you would never have identified it. So that's one of the main benefits there. See if doing targeted MRM assays on a UPLC triple quad, what adaptation to the instrument yields the most significant increase in sensitivity? Yeah, you can do microflow on modern UHPLCs for sure, or even higher flow. And it really just depends on if you can detect the thing that you're looking for. And the important things here are chromatographic resolution, narrowing the peak. So columns that use super small beads will always have a good resolution effect based on, like less than 2 micron particle beads versus columns that use 3 micron. Their peaks will be broader under the same chromatographic conditions. The most significant increase in sensitivity is often lowering the flow rate and moving to very small chromatographic diameter beads or longer columns that will improve your chromatographic resolution, or using orthogonal approaches like ion mobility or FAMES usually increases the signal to noise for MRM. SRM and PRM. So SRM is selected reaction monitoring on a triple quad, which means you're individually looking at the Q1 setting, so the precursor, and one product ion at a given time. And the instrument will always look at things in pairs, Q1 and the second quad, or sorry, Q1, Q3, and kind of go through the list. So in a case like what I'm showing in the Skyline setting, the 626, it looks at all of these product ions individually, and that's how a triple quad runs. Whereas with parallel reaction monitoring on an instrument like a TOF or an Orbitrap, it's generating the product ions all at the same time. And so in one scan, you get all of them. And so that's called parallel reaction monitoring. I see we're a minute over, and Lindsay is up next. I will be hanging out online to help continue answering questions. So I'm happy to do that, but I don't want to take away the Lindsay time for the next session.
Yeah, we've got a 15-minute break now. So feel free to get up, grab your coffee, grab a beer, depending on what time zone you're in. I won't judge. A little Irish cream in your coffee. We're all friends here. We'll stick around to answer questions. Reminder, try to make sure to use the Q&A in the chat. And we will get started again in about 15 minutes.
I'm happy to hang out during this 15 minutes. If anyone has questions, I can answer live.
Yeah, we've got lots of questions in the Q&A. Brendan and I have been cleaning up maybe some of the easier ones.
Okay.
But if you have any of them that you want to address.
All right, Brendan's on that.
And recaffeinated. We started at 8 a.m. over here on the West Coast. Oh, I think to unmute, I think I'm a host, so I can do that. Do you want to be my guinea pig, Lee Foster? Awesome. See, I just sent a request.
I accepted. Can you hear me?
Yes, we can. I did it.
Great. Hi, Lindsay. Yeah,
How's it going?
It's going all right. I'm home today with my little baby. He's kind of sick, but still here and still here and really interested in what you guys are doing today. So maybe since we're taking a break, I can ask a couple of questions.
Yeah, of course.
So I am moderately familiar with Skyline, but I've always been kind of confused by the verbiage. So maybe you could just help me understand, when you say transition, what is a transition?
You want me to do this?
You can do it. I learned it from you. It only seems appropriate.
Let me see if I might have a slide that. So a transition really is just talking about when you fragment your peptides, you start with a precursor and you wind up with a piece of the precursor or product ion. They used to be called parent-daughter. That is not the most up-to-date use of the terms. Now they're called precursor product, or transitions is the pair of those. On a triple quad, you analyze those in the first quad and the last quad. So usually Q1 and Q3, and that pair is called a transition. In PRM, it's pretty much the same thing, except they're not really quads. It's just the precursor and the product ion pair setting. So it's the transition from the whole to the piece.
Okay, understood. Thank you.
Mm-hmm.
I did have one more question, particularly on the data set that we were just looking at. So first of all, the data was beautiful. I don't know if you guys were using like purified proteins or what, but one thing that I noticed is that for some of those peaks, one of the Y ions was significantly more abundant than any of the other. And it's not something that I've regularly run into when I've been looking at data. So I'm wondering if you could speak to one of those ions being so much more prevalent and what that means for the data set.
Hang on. I might have examples of like this. So basically, what I see when I'm looking at data sets and I'm trying to figure out what signal is the one coming from that particular peptide. In the case of Skyline, it's pulling out all the product ions, right? Usually, when we digest with trypsin, we usually get a lysine or proline at the end, and there's often, lysine or arginine at the end, so a K or a R. Mm-hmm. If there's a proline before that K or R, then the intact peptide usually like fragments there just due to the nature of the proline, and that becomes one of the product ions that's generated with the least amount of energy, or it's like most quickly to fall apart versus all the others. And so the Y2 product ion, if it is that KP or KR, or sorry, PK or PR, you'll see this big signal because it's just the easiest to fall out. You shake it, it falls off, right? Okay. So, and that's also one of the reasons we don't normally like to use these small Y or small B ions, because they come from 2 amino acids. And if it's the Ys, we know that 50%-ish of the peptides have a lysine and 50% have an arginine. And so it's easy to miss a sign based on that. Usually the way I like to look at it, I'll just share my screen real quick and look at the Skyline file, is when we are looking. So for people who are wondering what Lee's talking about. Yeah, so this is a Y9, which is the most intense. I like to try to see that the other product ions are at least 10% of the biggest one. And sometimes you just don't get lucky with that. So in this case, I don't know why the Y9, it's something in here is fragmenting to generate that. But it is one of those things where you get what you get. and you try to see if it makes sense with the sequence of the peptide. But it's not always an ideal, nice distribution of all intense product ions.
Yep, understood. So in the scenario where you're talking about like a proline next to a lysine or an arginine, would that be addressed if you were to set the exclusionary criteria that you don't have any miscleavages?
It could, depending on if it generates a miscleavage or not. So usually the KP or RP would be the sequence where prolines after the lysine cleavage point, that would be a miscleavage product. Not if it's before the lysine or arginine.
Right. Okay. And just like my knee-jerk reaction when I see one that's very far above the others would be to exclude that particular ion, even though it's very high intensity. Is that something that you would suggest, or is there a particular benefit to having that particular ion?
So in my opinion, I would base it on what your needs are. If that is the only signal in the entire chromatogram you can see, there's no other peak that would be like a second best, then I might keep it. And if it's particularly the only peptide for that protein, I might keep it until I'm done with the method development and make my decision to keep it or axe it at the end. Because you might have some peptides that are looking inherently more stable, but, more equally distributed, but they might not be stable after doing run to run and having them sit in your autosampler vial for a while. Whereas one that has that weird distribution of like one super intense and the rest not, that may be like your best peptide you get. So I don't usually axe things early unless they look horrible and there are no other backup options.
Okay, thank you. I know Lindsay is going to get to more of this, but thank you for answering my questions in the interim.
Yeah, I'll definitely make sure to build in a bunch of time for some Q&A at the end so that we can go back and dive a little deeper into any of these sections that people thought were particularly interesting or relevant to their work. And